Yeah, we're definitely excited to have all of you here. It's great that there's so many interactions between kind of the commercial side as well as kind of academics. And so I changed the title slightly. It was, machine learning to cure cancer, and that might be a bit bold. I think that was Raj's little input. But basically, I'll talk about aspects of machine learning and how we look at biological data sets. I'll initially begin the talk, though. Let's give a little bit of background. But then basically, I want to kind of talk about some of the work that led up to the work related to cancer, and then basically how we've kind of are implementing it. But we really see ourselves, even though I'm in academics, it is almost like having a startup. It's almost like venture capitalists, which are the university, basically are funding us, my group, to do some interesting things. And they actually give you quite a bit of leeway to do so. And so I'll talk about kind of how we think about really ourselves kind of as a startup as well. And so basically, by the end of the talk, hopefully you'll get a sense of, when I say a feature of a cancer cell, hopefully you'll basically kind of be able to really maybe explain it to somebody else. And how do we actually look at cancer cells' response to a given drug, and how do we quantify that? And then once we understand how the data can be generated, and once we can quantify it, once we can put it into a really nice, maybe a Cassandra type of database, how do we then use the big data analytics? Now, what I want to convince you of today is the fact that we actively look at all of the types of algorithms that are heuristics that are used for big data analytics. And I'll talk about some of the kind of my influences as well as relates to these things. And so I got my doctoral degree in statistical genetics. And this is basically one of the, we've been using these kinds of large databases, millions of features, and trying to understand how basically we can create, I guess a statistical relationship between certain data points and a response. My initial work was looking at genome-wide association studies, looking at how human genetic variance actually impacts on our susceptibility to infections like TB and leprosy. And then I did a postdoc looking at basically how our immune response is actually impacted by our genetics. Previous to that, I actually had a few years at Harvard Medical School running this high-throughput DNA sequencing core for the Harvard Cancer Center. And as you see, previous to that, I looked at HIV phylogenetics, and previous to that, we looked at kind of the genetics of how tuberculosis virulence actually manifests. And I think a lot of academics actually go through various stages of kind of research. And I just kind of show this because basically, even though I was kind of hired to do genomics of infectious diseases, I'm coming back full circle, really, to some of the cancer work that I did up at Harvard. And so, again, I'm trying to convince you that the data sets that we have now and that actually are publicly available are really quite amenable to the kinds of big data analytics that you use every day. A lot of you use every day in your jobs. And we really are thinking about how we can create new heuristics, new clustering algorithms to actually understand, for example, if a cancer cell is going to respond to drug X or if it's better to give drug Y. We quantitate everything. We try to quantitate DNA information content. We try to understand about genetic mutations. So one of the main things that you kind of have to know is the whole central dogma of biology, which is DNA to RNA to protein. I try not to get into too much biology, but basically the idea that DNA encodes RNA, which then is a quantitative trait. So RNA itself, so let's say there's 23,000 genes. At a given time, let's say there's about 7,000 of those 23,000 genes actually expressed. And you have this amazing quantitative trait across the RNA expression, which changes drastically during cancer and changes drastically during infectious diseases. And so we care a lot about that. And these are data sets that we're formatting now, and I'll show you some of the plots later in the presentation, where we're really trying to link that gene expression to a trait, such as response to a given drug. And not only singly, but we're also looking at combinatorial effects of differential gene expression and how well a drug will work. Because the gene expression, the RNA, will then yield the protein abundance. And so without RNA expression, let's say a gene is shut off, then you will not have protein. And so we can make these basically very clear assignments of how these things work. When I say vectors of signaling pathways, and so the other thing that is really important in cancer is the fact that there tends to be a receptor on the outside of the cell, like epithelial growth factor receptor, which will then signal into the nucleus to have the cell grow. And not only do we have these really interesting graph nodes, but there's directionality, there's a vector to that. And we need to basically allow for the machine learning that we're going to do later to basically take into account those relationships. And so we quantify, we build algorithmic models, and then we refine those models, we test, and then finally we basically repeat. Oh, great. Oh, no problem. No, okay, thanks. Okay. It's on now. Okay, great. Is that better? It's a little bit easier to. Okay. And so we repeat this process, and we try to find these optimal models to build. So, really, biology really does fit into the kinds of work that you're doing. I show this slide because these are some of my influencers. So I went to school with Sean Gorley. We did our PhD at the same time at Oxford. He's now the founder and chief technology officer of Quid, which is a fantastic data analytics company, big data company, and they're trying to visualize these types of patterns within various, basically trying to organize all human knowledge. That's basically how he describes it. And I'll try to get him to actually come here to speak to you guys in the near future. I also know Chris Ahlberg. He's a Swede. I'm originally from Sweden, and I've talked to the VCs that actually funded his original company called Spotfire. Now, Chris Ahlberg is actually the founder and CEO of Recorded Future, and they were funded by In-Q-Tel and Google and others, and doing some phenomenal kind of really nice analytics across the web. And then finally, Alex Gray, he's a faculty member here at Georgia Tech, and doing some really nice support vector machine learning aspects, founder and chief technology officer of Skytree. So, when I do my science, when I do my actual algorithm development with my research group, we look at these guys. We look at what these guys are doing in terms of how they organize the data, how they try to visualize the data. And I want to give you a quick example of how this is actually how we try to create something new. And so in biology, things kind of like in business, kind of let's say they've been done a certain way for a very long time. Let's say for 40 years or so. We try to disrupt that process. We try to do something new. And I'm going to show you one example. It's not cancer related per se, but we want to show you how we actually create a new data structure and how we can process, let's say, genomic data, looking at bacteria and viruses in a slightly novel way. And so in the past, because of the limitations of hard drive space and RAM memory, there was real limitations on how much data we could store. And so we would tend to store smaller bits and try to condense it. And so, when you talk to infrastructure people, you try to have the smallest kind of data set available. So we decided to try something different. We decided to take the A's, C's, G's, and T's of DNA and actually create these invariant blocks, let's say, of a given size. In this case, I'm showing you something at the top of, let's say there's a 10 nucleotide stretch of DNA. And so we can actually take a very long thing. So the human genome is 3 billion of these letters long. And so we can actually take those and chop them up. And what we actually did was, in this work, we created a sparse matrix or sparse vector that encoded all of that information. And so maybe the simplest is to look at k size of 2. So k is the length of this of the nucleotides that we're storing. And so because there's 4 bases, a k of 2 would be 16 possible nucleotides. So let's say AA, AC, AG, AT, all the way down to TT. And those are 16 different spots. And so what we can do is actually break apart a piece of DNA and into those blocks and store all that information. Now, clearly 2 is not big enough, but once you get to K of 8 nucleotides or 10 or 12 in biological systems, it becomes very interesting. And then you can count the number of occurrences that you have of that given those bases. And what happens is, it turns out that once you have these sparse, it's kind of like a Yale sparse matrix format, very similar to that, you can actually start doing some really cool algorithms on top of that, whether you want to do orthogonal transformations of that data using PCA or multiple dimensional scaling. You can actually create a number of different visualization approaches to actually look at how similar two, let's say, genomes are. And so my group actually here at Tech, we think we're the first to actually have analyzed all known bacteria and viruses in this framework. We created a sparse matrix. And so normally you wouldn't do this. This is, it's not quite a terabyte of data, but it's close to that when you actually encode it because it's a sparse matrix. But you can actually do these algorithms on top of there, and you store them basically in a NoSQL type of database, and you actually do this analysis. And you can actually look at, for example, if you take the first 3 principal components, you can actually look at where they all fall. And the things that are closely related, it turns out, are very close together in that dimensional space. And then we said, well, what about if we use a discrete type of approach, discrete mathematics, like a Boolean transformation of that data? And again, you have to use these slightly larger block sizes, let's say of 14 or what have you. We store them. It turns out that it's a little bit hard to read here, but you can actually do these standard Boolean transformations of the data. So you can do XOR, you can do AND, or NAND, or any of these other functions. And it turns out that when you do this XOR function, you actually end up finding that things that are highly related, obviously, have a smaller distance between them. And we did this, and we actually were able to find that we found the same relationship. And so here's this kind of if you think of genome 1 here, genome 2 here, and so you could very easily, you can imagine with this sparse vector, you can actually do these transformations. And if you do the AND, you get this, you get the OR, and this is the XOR function that I was mentioning here. And so you see very specific distances between them. And then there's some math behind that. We'd look at Shannon entropy of these things, and the occupancy that we relate to, the differential occupancy we call tau. I'm not going to go into great detail there, but we can actually mathematically define these relationships. And so what happens is, within basically 2.5 years of being here at Tech, we've come up with these mathematical relationships of how speciation may actually work. So it looks like there's a power law distribution where if you do the rank order of the distances, and then it's a little bit hard because the numbers get small, so we actually do the negative log of the numbers. So the more similar those organisms are, whether it's in linear algebra space, in terms of principal component analysis, or whether it's using the XOR function where the similar species are actually have a smaller number associated, you do the negative log of that and you get this power law here. And so what happens here is these are the species, when you look at pairwise distances of let's say all bacteria in this case, so there's 2,500 data points plotted here, you actually see that the ones that have a close pairwise distance together, again after you negative log it, those are the ones that actually are very similar species. So we do the Z transformation here, and so we take Z is 2 or greater, and it turns out that these actually are related. And these have been defined previously to be highly related in terms of species here. And so we define within all of these bacteria actually things, whether we use linear algebra, and we just plot the discrete math score next to linear algebra score, and we get this really nice R-squared relationship, meaning that both of these techniques actually do a really good job of finding highly related species in this kind of information space. And so, I went through that a little bit quickly, but I just wanted to kind of mention that the way that you format the data is actually really important, and it can be useful. I didn't go to MIT, but I've listened to the engineering lectures, and they talk about layers of abstraction in engineering. And so, is the layer of abstraction that you create, is it useful? Is the kind of the kind of new format to look at this thing useful? And we argue that it is, because even though it takes a lot of hard drive space and RAM space to actually store these sparse matrix of these biological data sets, the ease of use and the actual computational capability of being able to look at two divergent species becomes really useful. And so we can do this. We can actually plot all of them together and look at the ontological relationships between, let's say, all bacteria. And so this was not possible before, because you had to have a certain amount of similarity to the species to be able to do the alignments. I should have mentioned that earlier. But basically, and so, this is the kind of really cool types of things that actually could probably be used in other fields as well. And so this is not submitted, so this is kind of the first time this is being shown. Here's an example of looking at these very divergent viral species and looking at how similar they actually are. And so it's impossible to do normal types of algorithms to actually look at the similarity, but here we actually can see the larger these lines, the more similar they are. And so we actually can find relationships, these cryptic relationships that have not been defined before. And so this is one of the first papers that we published from the lab, and so it's in the journal Bioinformatics, super geeky, but basically it's on SourceForge as well. This is called KeyAnalyse, and so this just came out in the last couple of weeks here. And basically, it's a kind of an optimized Java program to do this type of analysis. And we're hoping to basically spread the word about this, because it's a very efficient way of looking at this type of data abstraction that's being used for genomics quite a lot now. And so even though I'm kind of in the School of Biology, we're doing a lot more kind of the algorithm development than maybe even the faculty expected, but they're quite happy about it nonetheless. And so, so as relates to the background of cancer itself, well, it turns out that we can use the same types of tools, the same types of ways of looking at this abstract data for cancer as well. And so the backstory behind this was that John McDonald, one of the heads of basically the Cancer Institute here at Tech, he was the former head of genetics at UGA for many years. He basically recruited me and said, well, you're in the genomic space. I want to basically work on the cancer problem with you. And so we applied for a grant to this private funding agency, and they basically, we thought we were just writing a short white paper to then write the formal grant, but they basically just handed us over nearly $1 million to actually do this after just a 3-page write-up. But the idea right now is that in genomics, it's really evolved in such a way that in about 10 years' time, or even 5 years' time, it's going to be completely inconceivable that we don't quantitate and quantify a tumor for a given cancer patient. It's going to seem archaic that we don't know the actual DNA. And a lot of companies are doing the DNA space. But more importantly, as I mentioned, in order to get protein, you need the RNA. And so the RNA space becomes very, very important as well. And so we basically proposed this model to the Rising Tide Foundation, John McDonald and I, and we said that, well, everyone's looking at the DNA space now. Why don't we look at both the DNA and the RNA space and other aspects of the tumor? And not only that, why don't we look at the tumor basically at different time points itself and seeing how the tumor actually mutates. And so what normally occurs within cancer itself is that a DNA mutation, some type of change will occur, and that will either cause certain genes to be more functional or cause certain genes to be completely shut down. And what happens, though, is that in cancer, it's not only just a DNA mutation that can induce a change in RNA, but you can also have what we call epigenetic modification. So modification to the DNA structurally that prevents the RNA from being made properly. And so now it turns out, and you know, the backstory a little bit too is that you know, cancer has historically just been named based on where you find the solid tumor. And so you have breast cancer, prostate cancer, lung cancer. And these are not really good definitions. These don't do justice to the complexity of what's actually happened to cause these cells to grow out of control. And so genomics in this field is transforming that because it gives us a very defined classification and even the ability to understand why the tumor is doing what it's doing, and potentially the ability to really inform what drug to actually give for a given patient. And so what we're basically defining or working to define and you know, using machine learning to actually put on top of that is to really understand the features of the cancers that actually will link to this, and basically, you know, how do we actually optimize based on that? And so when I talk about features, okay, so you know, we mentioned DNA and how it basically is a precursor to RNA. Well, so it turns out that DNA, this is kind of the double helix structure, which was really defined in 1953 by Jim Watson and Francis Crick, basically looks like this. And so each of these stacked bases are basically encode information. So you have AT and GCs. And during cancer, for example, I'll show you a slide in a little bit, but certain cancers are really defined by a very high mutational load. So a lot of these changes are occurring for the cancer, such as lung cancer or melanoma, two cancers in which, you know, so UV light, for example, can damage cells. And so you have, let's say, two thymines next to each other basically chemically link up, and then you can induce a mutation if you don't remove those adjuncts. And so we have DNA repair mechanisms, but they don't always work. And so cancers basically evolve at the DNA level, but then that can impact on the RNA, and it can impact on the protein itself. So if this piece of DNA encodes one of the exons, these are portions of the RNA that code into protein, then you can get a change, and you can actually get an upregulation of the function of that protein itself. So it's not only a single nucleotide change. You know, you probably heard about this quite a bit. It turns out that during cancer, whole bits of the DNA can actually just be amplified. So you could have 3, 4, or more copies, basically of a portion of a chromosome. A lot of strange things happen in cancer. You can get insertion deletions. You can get these inversions and translocations, and a lot of very, very unusual things that occur because really cancer cells stop being able to repair the type of damage that healthy cells tend to be able to repair, and they don't shut off the way that they should. So when I say RNA changes, it turns out that we can look at these RNA changes for a given cell as relates to kind of normal. So for prostate cancer, like you'd have a cell and you would see changes in a few of those 23,000 genes, or many of those 23, that basically reflect how the tumor is actually able to grow. And so you can get mutations, but also, like I mentioned, these non-DNA changing events that can actually shut down a certain gene, and this happens quite a bit. And so, you know, basically, you know, it turns out that the RNA changes may be actually quite a bit more important to understand how a drug will work, even above and beyond the DNA changes. And we have preliminary data to back that up. And so when you look at the FDA-approved cancer drugs, there's really, I see kind of two classes. One class is a general class where you basically have, it basically will kill any rapidly dividing cell. what's happening, what's transforming cancer, though, is that you have the second class, a very defined small molecule, usually, that will basically bind to one or a few different proteins. And it's a targeted therapy. And so the idea is that, for example, if you have a mutation in, let's say, one of the proteins, and a lot of these are kinase inhibitors. So kinases have an interesting property. They're able to modify other proteins with a negatively charged molecule. And sometimes these kinases, these things that add this phosphate, this negatively charged phosphate to proteins, actually are overactive. And you basically want to reduce the amount of that activity. And these kinase inhibitors are very good at that. And so now with genomics and molecular medicine, we can define very specific changes in not only the DNA, but also the RNA level, which will allow for direct targeting of these pathways that are aberrantly upregulated. And that's basically a very exciting time. And you'll hear more and more about these things. And while I'm kind of talking about this topic as well, is that it's pretty clear now, if you're familiar with the initial HIV drugs that were given to HIV patients, it turns out that those single drug regimens that targeted, let's say, the enzyme that reproduced the virus, the polymerases, it turns out that mutations would occur in the virus very quickly, months even after the drug was given. And this was a very hard thing to deal with initially because there weren't many HIV drugs initially. And so a second drug, a second class of drug, let's say a protease inhibitor, was added to that repertoire. And now it would take, let's say, a year for resistance to develop. Well, it turns out with triple or quadruple therapy, resistance just doesn't develop. And again, what's going to seem very archaic within the cancer field is this idea of giving a single molecule. Because cancers inherently are able to mutate very quickly. They've kind of lost the ability to repair themselves. And so what's going to happen is we're going to have small molecules. It's going to be at least triple therapy or quadruple therapy before we can get closer to, I don't want to say a cure, but both John McDonald and I really believe that within, well, it takes a long time to approve these drugs, and so that's the only limiting factor. If we could decrease the time to approval, that would help a lot. But basically, within our lifespan, cancer could become a treatable condition like HIV is now, with the right knowledge about how the cancer is actually, the genetic state, both on the DNA and the RNA level, and with many more small molecules that can target additional kinases. Okay, so this is not my work here, but this is some of John McDonald's work, actually with Alex Gray, who's now the Chief Technology Officer of Skytree. Here's John here, and here's Alex Gray here. And I just want to kind of bring this up because there's another way in which machine learning is actually used, kind of a big data application within the realm of cancer biology, and kind of get you to sense that we really try to quantify these biological systems, and we try to understand how we can analyze that data in interesting ways. And so this is one way to do so. If you want to diagnose ovarian cancer, sometimes it's really hard to see any changes really that are occurring in these patients. And so what this study did was to actually look at serum, look at just the blood. It's a very non-invasive test. You take some serum out from a patient. I'm not sure if they're a patient yet. And the idea is to take the complex metabolites that are in your blood anyway and to analyze them using mass spectrometry here. And so you end up getting these really quite interesting complex data. And so what you can do there is So what Alex Gray and Skytree now are exceedingly good at support vector types of algorithms. And so, they tried many different ones, but it turned out the support vector was actually the most useful. And so you can actually read in these quite complex data streams in, and just like you would do for other applications, you could actually start to train it. And then when you actually look at real data, you can test to see how well you do. And it turns out they do very, very well. So people that actually are knowledgeable about this, they actually had very impressive sensitivity and specificity to actually detect the cancer metabolites within blood in a non-invasive way versus the healthy individuals. They reached out to Tech Transfer about this, and were like, well, you can't really patent this. And I was just a bit taken aback because this is a very powerful method to non-invasively actually detect cancer. And just because you can't necessarily patent the actual metabolites themselves, there's a Supreme Court case that came down with Myriad Genetics, and basically, and this is actually good for the field, saying that these biological systems, you can't patent a particular a trend in the data. You can't patent particular metabolites necessarily, but still, this is a very kind of exciting aspect of taking very complex biological data and making inferences using the types of algorithms that are commonly used for other types of data. So, once the data is formatted, then there's some really, with your background, you could actually start having fun with these kinds of things as well. Okay, so here's just a couple of slides. There's a really nice paper published in Nature August of last year that took from the National Cancer Institute as many samples as they can get their hands on, really, and looked at, for example, DNA mutation within those samples. And so we try to find trends in the data. And one of the trends that stuck out, really, was the fact that here, again, we tend to log scale things to make it a little bit easier to see. And so this is on a log scale. But these are the number of mutations found for different types of cancers. So it turns out the cancer here, well, here's one that you might have heard of, this ALL, which is one of the leukemias, one of the B-cell leukemias. They tend to have a very small number of actual average number of mutations here. And so it turns out that most of the actual occurrences are not because of a single nucleotide mutation. It's actually this translocation that puts a very strong, what we call promoter element onto, let's say, one of the key oncogenic drivers like MYC. And so there's not many changes that have to occur for that cancer, ALL or AML, to occur, or CLL. On the other end of the spectrum, melanoma, which is a skin cancer that's caused by UV damage of your skin cells, basically there's tons of mutations here. And then for the lung cancers themselves, so because of smoking and other environmental hazards, you tend to have a lot of mutations. And so even without even knowing necessarily what type of cancer is, I mean, that's actually kind of a useful thing. So you could definitely tell these two apart using machine learning approaches just by looking at the number of mutations. And we call this analysis tumor normal analysis. And so we basically have germline, which is kind of what all our cells should have, and then we have somatic mutations. That's just some of the terminology. Somatic are the ones that occur later later in life. And so we have some patterns there. And so again, kind of as it relates to machine learning, in this Nature paper, try to define a series of different, say 21 signatures that relate to specific types of cancers. And so we can start to try to define them in that way. And then here, these are very kind of common mutations that are occurring. Whenever you see these types of things, let's say here's a KRAS glycine to valine change. So the glycine 12 to valine. So it's the 12th amino acid in that actual protein, and this is a very quite frequent mutation because it helps activate this thing, which adds these negatively charged groups to other proteins and helps drive the cells to grow. Another common one here is this BRAF. Again, another kinase adds these negatively charged groups onto these other molecules. And when I talk about signaling, so you're basically signaling from the outside of the cell down into the nucleus to drive the cancer to start growing. This is the kind of the vector that we're talking about in terms of the signaling. The direction of the pathway is from the cytosol into the nucleus. And so here's another really nice paper from a systems biology journal that was published last year. And this is not our own work, but it was very nicely done. And I just wanted to show you a demonstration of how this could be done correctly. And so this is what we call this vector from the outside of the cell into the nucleus. And so this is the outside of the cell here. Here's this epidermal growth factor, which is commonly mutated. There's common mutations to basically leave it on all the time. It should only be turned on in healthy cells when this red molecule binds it. So one of the common things in cancer, and this is what we need to know for therapeutics, is that a change will occur to it's basically like pushing the gas pedal down, where that car is just going to go. And when you take your foot off the gas, the car should slow down, but these are changed, and so that gas pedal is pushed down all the way. And then it goes through this RAF. Remember the BRAF that I mentioned in the other slide? And these are called kinase pathways. So these are adding these negatively charged molecules, these phosphates, onto the downstream steps, and it basically potentiates this down. And so what these little squiggly lines here show are these specific inhibitors of those given pathways. And this is kind of where, I'm not going to go into too much detail, but in that initial slide, we talked about how we build these mathematical models of how the signaling works. So we have this in the literature, and we actually try to see how it works. But it turns out that it's not always right. And so we have to rebuild our models and create basically the correct models. And these models are getting better and better and better every day. But this is kind of what I wanted to show you here, is that it turns out that a single inhibitor doesn't always work. In fact, sometimes you have to have these combinations of drugs to actually allow the process to work properly. And so you can have different combinations give different effects, and basically, we can learn based on these things. And so we can empirically try this, and we can actually understand the feature space of a given cell itself, and the ability for example, the number of cells to basically, you can see here that either of these drugs by themselves do not work, but if you give both in combination, the cells really stop growing. Here, neither of these work, but once you give this in combination, these two inhibitors of the EGFR and, let's say, BRAF here, so these are the kinases, and these are kinase inhibitors, and you can target it and you can turn off the cell in that way. Not only can you turn it off, but there's a lot less likelihood that you'll actually get a mutation, which will then basically help to inactivate these things. And so these are the kinds of things that we need to basically fit into the models. And so basically this is kind of the exciting time now. Just to mention a couple of the companies, it turns out that Foundation Medicine, it was basically a very new company founded by some of my colleagues from Harvard, including Levi Garraway, Eric Lander, and others. It just actually went public this last year already, and so there's a lot of excitement behind looking at kind of the genomics of these tumors and understanding how they actually will work. And so again, this is only on the DNA side. They're not doing the more important RNA analysis. And then there's a company also up in Boston called N of 1, and kind of think of clinical trials that have thousands of people. now they're talking about personalized medicine, so the N of one, basically the one patient at a time. And they are also, again, using the DNA mutations, trying to build these mathematical models and try to give patients data about their actual tumors themselves. And just so you know, I mean, this is a very exciting time within this space, and we're actually, both John McDonald and I are kind of seeing this as kind of an opportunity to have Atlanta actually be able to contribute to this field. So it does not just have to be the Bostons and the San Frans. We can kind of see how this is going to basically play out in the very near future. Be able to basically capture the feature space, but then be able to use our own algorithms across that data. And so this is the, you may have seen these commercials. It's the Cancer Treatment Centers of America. They're trying to implement this type of personalized medicine approach. And so this is, the Rising Tide Foundation is kind of their nonprofit arm. And so they're the ones that sponsored us for this research. And we're kind of building these models now. And so I'll just take the last bit of this talk to basically discuss like how we're kind of building this. Oh, another, I wanted to mention this too, another kind of an exciting initiative is the idea that data is becoming, these systems are basically set up now to help algorithms become as good as possible, right? So you have these Synapse and others are teaming up with these various organizations in cancer to optimize let's say the DNA calling as well as the RNA analysis. And so there's a lot of excitement there where you can just have open data and basically create kind of challenges or competitions to do this as efficiently as possible. And there's definitely a lot of opportunity for any of you to kind of get involved in these types of initiatives. And I like that Raj and Travis have kind of done this for CF previously, and I could see kind of a cancer type of hackathon could also be really interesting. I've been reading this Eric Rice book, The Lean Startup, and kind of seeing how these strategies actually work. And they talk about this minimal viable product, this idea that you build quickly, you code quickly, and you see what works, and actually basically determine the key features, and then pivot as you actually want to change the functionality and kind of see where the model is going to go. I don't have a slide about this, but so I've become the director of kind of the genomics core, the high throughput DNA sequencing core here at Tech, and I see the actual prices being that it costs to analyze the DNA and the RNA changes. Foundation Medicine approached Northside Hospital and said, well, $7,000 per patient is our kind of a price point, which is on sale. Well, it turns out they analyzed 400 genes and they use N of 1 as their kind of analysis group. The price probably of the actual commodity of DNA sequencing we could probably get down to about $600 or so. And the algorithms to analyze that can actually be done in an open source way, so people can actually have access to the mutations in the RNA space. So it's almost like a social necessity that more initiatives actually are out there to allow for this analysis. Certainly, if one of my family members were to get sick, or to acquire cancer, I would want to know very quickly. So I guess what I'm trying to say to you is that in the very near future, you could pay out of pocket $600 to do this type of analysis that would normally cost a lot more, because these aspects of the insurance companies charge back and everything else. It's a similar case to an MRI. So my father had to get an MRI, and so we got a quote from, I'm not going to name which hospital system, but a local hospital system, and it's going to be $8,000. I was like, that sounds like a lot for an MRI, because it was going to be $1,800 out of pocket for him. And so I called around, and I found another place with the exact same machine that charged $400 with a $35 copay. And so this is going to become the same way, I promise you. And you're going to be able to shop around for these types of things. And I would almost rather pay the $400 out of pocket rather than the $1,800, copay to do it another. So medicine is changing, and these it's a free market system, and the market will decide what prices can actually be paid. And so the beauty too with cancer research right now is there's so much data available. So the National Institutes of Health, in large part due to Francis Collins, who is now the director of the National Institutes of Health, and they spend billions every year. Well, he was a genomics person, so he basically helped the U.S. effort to sequence the human genome, which was completed kind of in a celebration in April 2003, and now it's 10 years on already. But the idea is that basically, 11 now, basically that the government will pay for it, but that data becomes available to registered parties. Anybody in an academic setting or even companies can apply. You just have to fill out the paperwork and actually get the data. So it's really exciting. So we have all of these publicly available large data sets. And so kind of using that lean startup model, we're kind of doing a very personalized approach for the ovarian cancer patients that we actually are looking at, John McDonald and I. And so we can actually start to basically come up with an optimized approach for a very small number of individuals first, but then that can scale later once the software is actually built. And so we can gather all this stuff effectively. And so, we have this internal process now that we're kind of building. And I just want to show you a little bit of the kind of data that we're iterating. I mean, it's still very early days, but this is one of my PhD students, Pete Adwana, who actually helped to wrote most of the code for the KeyAnalyse that I showed you earlier. And basically now we're making correlations with RNA expression, which we find to be a more accurate predictor of whether a drug is going to work than just the DNA changes. And so now, as we saw this EGFR here, and so alertinib basically binds EGFR, and we have this ERBB3 here on this other axis. And so it turns out that on a log scale again, the farther on the x-axis here or the farther on the y-axis means more of that RNA, which means more of that protein. And it's interesting because you actually see that the larger the dots here, the more effective the drug is for that given individual tumor, individual patient, let's say. This happens to be in vitro work, but the patients could be presented in a very similar way. And it shows that this quadrant here corresponds to the actual the individuals that respond to the drug. And so you can actually look globally across all individuals that have been that have this kind of data and put like we do with LDL levels or something, you can put yourself on that distribution of all people and see where your LDL levels are. Same thing can be done for these things. And so we're actually generating, we're iterating on this. It took about 5 days of a high memory system to do this, but these are the lists. And so the Q1 again is this top right quadrant where if you're high for these two, both of these genes here, then it's very likely that then those people fall into the group of responders. And so now we're finding ways to actually, so again, this is an abstraction of the data. And you basically, you abstract and you actually, again, so this is one combination. So there's 23,000 genes, and so there's more than 400,000 combinations that you can try with this. And so we're, this is just one of many approaches that we're looking at, but we're looking at the kind of combinatorial effects of multiple, the expression of multiple genes, and seeing how well this does to actually predict a drug response. So that's another way to do it. So we have these mathematical models of known proteins and known mutations which will affect the drug, and so we'll use those in the models. But then we're coming up with new ways to visualize the data, as we've actually, as I mentioned. So I'll just spend the last couple of moments just kind of showing what we've built so far for this effort, and we're calling it basically like 3 modules. This is how we kind of defined it. And so the first is this DNA feature space. how do we define that? The second is the RNA space, and then the third module is this using machine learning to actually interpret this data. And so, we basically just kind of schematically, like any initiative, you kind of map out how this is going to work. Basically, you take the DNA from all genes, all the protein coding regions, and so it turns out that this is about 1% of the entire 1 billion bases, so it makes it easier to do, about 30 megabases. So we look at all of those. Here's another kind of interesting thing. So you basically look at the difference between mutations that are actually driver mutations, which we think are important for the actual oncogenic process, the actual having the cells basically have the gas pedal all the way down, versus what we call passenger mutations, which are kind of just byproducts of the DNA damage from, let's say, the smoking. There's a lot of changes that are occurring that don't necessarily link into changing the ability for those cells to grow. But the way that the driver mutations are actually identified now, there's a really nice random forest approach. Turns out it's written in Fortran 90, which is kind of old school, but the Cartin Labs from Johns Hopkins came up with, which is basically using all of the feature space of known mutations that are driving the cancer, and it's basically trying to, and then you basically you train it based on inducing 2,000 passenger mutations, random mutations that are not the driver, and you run that through the random forest approach a couple times, and then we put our actual samples on there of unknowns and define to the best of our ability which ones are actually linked to being drivers themselves. So there are approaches out there that are kind of open source or available. And so we used to use the Proton machine for this. We now have this new higher capacity machine called the Illumina HiSeq machine, which is just installed in my lab, and so now we're basically creating these various flowcharts. So it's very similar to what a company would do. And here's kind of CHASM is the software that we're using now for the driver mutations, and but we have a number of other modules that we've kind of built, and we're in the process of publishing these and just making it completely open source. So I think I don't want to go much further here. I just had a couple of slides that may just kind of summarize this a little bit. I don't want to go into too much detail. So, and I'll have some time for questions as well. But the idea is that we're really in the age of quantifying these biological processes. We're in the age of really understanding very specifically, minutely, down to each single base pair of each protein in a tumor, which one is changing, what's going on in terms of the RNA. There's just a paper that came out in New England Journal of Medicine in the last 2 or 3 weeks showing that you could actually identify colon cancer non-invasively now instead of having to use a colonoscopy based on DNA features called methylation of certain promoters. And so there's a company now that has a test to do this. But it's just interesting because as that Myriad case came down with the Supreme Court, it turns out that nobody has ownership over that test. And so we could do that internally here. You could actually do the exact same, I mean, it wouldn't be FDA approved quite yet, but you could actually get it eventually approved where you can identify with amazing precision through a non-invasive test the presence of a tumor in the intestines, and so this quantification, and this is only going to happen faster and faster and faster, and so we can really quantify things now. One thing that I actually heard first in high school back in in the day, and it still rings true today. And I'll give you a brief just anecdote about it. It turns out that cancers basically upregulate genes that just shouldn't be on in the adult. A lot of the genes, for example, relate to placental development. Why are males expressing genes that relate to placenta? So it turns out that cancers are able to, once the DNA damage starts happening, the RNA starts being expressed in ways that it shouldn't, and so all these mechanisms are turned on later in life. I think there's a gene called MST1. Again, this was just published about a week ago in the journal Nature, which is one of the best ones. And it turns out that this gene is not required in healthy cells, but it turns out cancer cells are very stressed, and they need to be able to basically repair certain building blocks of the DNA before the cells grow. And so it turns out that this group in Sweden found this at the Karolinska. Basically, it's a Nobel Prize, worthy discovery, saying that if you knock out this one single gene, numerous different cancer types actually end up, the cancer cells end up getting very sick and die, in fact. And so I guess what I'm saying is that we are iterating so quickly now. The data is available for, just like, kind of the page rank that, Sergey and Larry came up with. The same data is available to all of us now, and it's kind of our mission in part to explore that data, to find themes in that data, find patterns in that data, use machine learning, use big data types of analytics to find those patterns, and really start not only diagnosing but also being able to treat these cancers. So, all right, I'll end it there. Thanks, guys. Yeah. Let's see. Sorry, I went through it very quickly.
I'm wondering if you start out with normal cell transcription regulatory networks to make your guesses on which gene pairs, or is cancer it's already too far away from that, so it doesn't help you?
Yeah, I mean, we certainly have those normal transcriptional motifs. Basically patterns. It's interesting because the work that I was doing in Oxford for my postdoc actually relates to how normal cells react after an immune insult, for example. And it turns out that about 30% of the genes change. So dynamic changes are common. It's just that in cancer, there's a dysregulation of the things related to how the cell cycle is driven. So we do a lot of the kind of normal analysis as well, but the story is kind of more interesting and complex than that. And so one of the things that we've started doing now in the lab is actually thinking about the physiological levels of a given RNA and protein. And you can think of it as low, medium, or high. And at a certain point, the low is defined as not enough RNA to make the protein itself. And those perturbations are the things. So we can think about it. So we have those the normal as well, but it's it can be useful, but not in all cases, I guess. I don't know if I explained that well, but basically those models can be built and are being built. Yeah, in the back. Yeah.
I was wondering, with that sample that you had on the mutations for the various types of cancer, did the mutations differ based off of the individual's DNA? And what I mean by that is that recently Cal Berkeley came out with a study through anthropology of evolution of man. And when they looked at in the DNA, the ratio of whoever had the most, anyhow, like,
Yeah, I mean, that's a great question, actually. So, if you look at population genetics, there definitely are certain common variants that protect or have an association with protection against acquiring a certain cancer. And it can be, I don't know if it could be a selection or if it could just be by random chance, certain, let's say, people in Italy versus in China could have a different allele frequency for that protective variant. And so one way that could manifest potentially is that, let's say there's a variant that relates to how well DNA is repaired or something like that. Then you could have certainly people with that variant that's distributed. A lot of the initial work that I was doing is actually looking at how, the immune response actually was controlled by different variants. And it turned out that one of those studies related to a mutation in a protein called toll-like receptor 1. And that mutation actually allowed certain ethnic groups to actually be protected against this infection. And in the out-of-African expansion, that variant became actually the predominant allele in Europeans and people in this room. And so the mutation actually caused a hypofunctional protein, not able to respond as well to the pathogen, but for some reason that was protective. And so yeah, there are differences in, and some of those are due to selection in the human across the different ethnic groups. And so that could occur in cancer as well. There's not as many well-documented examples of selection related to that because it's such a deleterious disease, but maybe for a cancer that has a later onset or earlier onset, that could be possible. But yeah, it's interesting. I mean, that brings up the point that the variants that we normally have, they can actually help regulate our susceptibility to various diseases, including cancer. Our diet becomes incredibly important, and one of the questions that was brought up recently is, well, why does obesity actually help relate to cancer? Why are cancer rates higher in people with higher body mass index? And it may be a little bit because of this example of how inflammation can impact on healthy cells. Especially body mass around the waist can actually create a very immune activated state, and there's been a couple of papers showing that immune activation of these cells can actually impact on driving certain tumors to form. But yeah, there's some interesting interrelationships between genetics, common variants, environment, and these things, and they're all kind of form this Venn diagram of risk factors.
I haven't looked in depth of all the publicly available gene expression data sets, but do they have a lot that are also like time differential expression, or most of them?
So that's a great point. I mean, that more of those need to be generated, more of those temporal time things. So there's more than 1 million data sets available, and some of them have that temporal aspect. But for example, for seeing how well a drug works, we really need to see pre and post drug to see what's happening to the RNA, what type of compensation is occurring. And those things are starting to happen. We need to know before and after resistance to a given drug, for example, and those things are being collected now. It's just, the price of DNA sequencing, the price of analyzing these things is going down drastically, and so we're going to have a lot more opportunities to look for those types of, get that more rich data in very soon as well. And yeah, we're hoping to make kind of Atlanta a hotbed of that type of analysis. Yeah.
Okay.
Yeah, excuse me, I have 2 questions, if you will. Going back to your KeyAnalyse protocol that you have been developing, you're looking there at viral variants and you're looking at bacterial variations, and essentially establishing a phylogenetic framework within those groups. Is there any interest in moving that to a larger phylogenetic analysis?
So I mean that's the kind of the thing. So we basically call it, I guess it's clustering. I guess it's just brute force information content similarities. And so we kind of don't like to call it phylogenetic because it doesn't necessarily, it won't fit the other trees because there's been a lot of what we call horizontal gene transfer. So we're capturing that, but other people try to exclude that because they look at very conserved DNA areas. We're definitely, it turns out that this type of analysis is being used for a lot of different things, including gut microbial studies, looking at different bacteria that are in our guts. And so we hope that KeyAnalyse will hopefully be extended to be used for that. And so right now, a lot of people are using this software from Carnegie Mellon called Jellyfish, and so we're trying to basically compete with that, and the paper shows that we compete well with that approach. But we're actually even thinking about extending it into you could even do it for these RNA studies, actually. You can look at the raw reads coming off of the machines and then doing a real-time analysis. There's some normalization that has to occur for that to work well, but I think just building something from the ground up, it's kind of nice to actually start to just put it onto different problems. And I think that certainly for what we call metagenomics or clinical metagenomics, it can be used out of the box. But we're kind of coming up with other algorithms. One of the other professors here at Tech, I showed him this sparse matrix format, and he really got it. So he came up with this other algorithm to work on top of it, basically, to cluster different species together, and he does a very good job too. So it's fun to put these data structures out there to look to have other people analyze. And I should mention too that we created we also created a kind of a matrix format as well, because there's a lot of matrix calculations that can be done that maybe isn't available for just a vector as a linear vector. So we kind of hope that these technologies will be kind of extended upon a little bit like the open source movements and various other initiatives, obviously.
Yeah.
Given the fact that you're saying there's likely to be multiple compounds that are needed for a single cancer, and there's multiple types of cancers with multiple types of pathways, do you foresee a better methodology than maybe the conventional FDA methodology of one compound being effective for treating one type of thing?
Yeah, absolutely. But now we have the justification for giving a certain compound. So it turns out that just because it's prostate cancer or breast cancer, we can identify pathways or a given small molecule that will work for both, right? And so that's happening more and more. And the FDA and the National Cancer Institute is very interested in these cases where, for the vast majority of people, the drug doesn't work, but it cures the 2 or 3 people in that trial. And so they're really interested in actually understanding the genetic makeup of those individuals, of that tumor itself. It's changing. I mean, double therapy is starting to, there's almost all the trials involve double therapy. It's going to be triple and quadruple. It's going to be next. And I think we'll see the success rates move, to become much, much better after that occurs. Because I think, it's been 50 years and we haven't really gone very far with cancer yet. And so, the government has to be agile as well in changing the way that these trials actually work. And I think it's going to happen. So the nice thing now is we can actually test it. We could take some of the tumor out and actually test it in vitro and show that it doesn't cause toxicity for the healthy cells, but it reduces the growth of the tumors. And, that maybe medicine will get that specific in the near future for these N of 1 individuals, where you can actually give justification there. So,
yeah. I had a second question. What you're doing, you're calling statistical genetics or genomics, but there's another field of it in animal breeding, quantitative genetics. And I'm wondering if you could speak to any relationship between them or any.
Yeah, I mean, we use I mean, I kind of use them a little bit interchangeably because a lot of the studies that we've done are quantitative loci mapping in human samples. So yeah, I mean, we kind of just under the umbrella of genomics. I think they both quantitative trait analysis as well as statistical genetics kind of fits under that umbrella, that broader umbrella. But yeah, I mean, that's some of the more recent papers have actually used those approaches to find some really interesting interactions. We used to take, let's say, all blood and look at the RNA expression there. So in a recent paper, we took the B cells and the macrophages separately, the monocytes separately, and with the same genetic background, you'll actually, because they have the same genetics, they actually have different gene expression. For example, a mutation in a promoter in a gene that's turned off just won't have an effect, but in the cell that has that gene, let's say the lysozyme gene was one example, a SNP will drastically change the amount of that gene. It could even be that a mutation will increase it in the B cells and decrease it in the monocytes. And these interesting relationships can be found by, again, getting more precise about what cell we look at and other things. And so, medicine is getting much more precise now. We're getting into very, a very precise kind of analytics off of smaller cell groups, specific cell types. Yeah. Oh yeah, just one more, and then we gotta wrap up. Yeah, go ahead.
There's extra benefit into looking into additional data to expression, like a graph kernel on protein interaction.
Absolutely, no, exactly, and so yes, it's just that the RNA data is just more available now. But if we had the protein data, it would be even better. We can add those things onto there, absolutely, and it's just amazing with all these kind of open source analytics tools too. We can just start, and so Alex Gray, for example, I mean, he, they have about 10 different. Support vectors is one of many approaches that they try, but so you basically iterate and you see what does the best and utilize what's best. But yeah, we will get that kind of data too very soon. All right. Okay. Exactly.
I want to thank Dr. Vannberg.
Okay. Thanks. Yeah. Thanks.
Thank you so much.
Thanks.